For this assignment, I'll be discussing two lesser-known pathogens, Mycobacterium ulcerans, which cause burley ulcers, and Coxiella burnetii, which causes Q fever. Mycobacterium ulcerans produce a toxin known as mycolactone. While not traditionally considered an adherence factor, mycolactone plays an important role in immunomodulation, which might indirectly facilitate adherence and colonization by suppressing the host's immune response. At the molecular level, mycolactone diffuses readily across cell membranes due to its lipophilic nature and binds to the host cell protein Sec61. This interaction leads to the inhibition of the translocation of proteins into the endoplasmic reticulum, which is fundamental for normal cell function and immune signaling. This bacterium has a unique capability to invade the host through skin breaches and then spread without eliciting a significant immune response. The bacteria produces mycolactone, which causes programmed cell death in cells, aiding the bacteria in penetrating deeper tissue. Once established, Mycobacterium ulcerans persist in the subcutaneous fat, where mycolactone causes extensive tissue damage and inhibits the immune response, creating an environment that allows the bacteria to colonize effectively. This promotes the development of large ulcers. This is achieved through its cytotoxic effects on fibroblasts and other skin cells. Mycolactone reduces the production of extracellular matrix components and growth factors. This delays wound healing and leads to more severe tissue damage. Mycolactone also acts as an immunosuppressive agent, enabling the pathogen to evade host defenses. It inhibits the production of pro-inflammatory cytokines and can cause cell cycle arrest in host cells, undermining the host's ability to mount an effective immune response. The bacteria typically exit the host through an open ulcerative lesion, allowing the bacteria to spread to new hosts indirectly through environmental contact and contaminated water sources. The toxin produced leads to the characteristic signs of the burley ulcer, including extensive tissue damage, immune suppression, and the formation of large ulcers on the skin. Coxiella burnetii adheres to and enters host cells through receptor-mediated phagocytosis. The bacterium targets mononuclear phagocytes and can survive in macrophages by inhibiting lysosomal fusion. It penetrates the host barriers by exploiting the host's endocytic pathways. Once inside, it resides within a modified phagolysosome where it can safely replicate. Coxiella burnetii uses multiple strategies to survive inside the normally destructive phagolysosome of macrophages. It alters its surrounding phagolysosome into a specialized compartment called the Coxiella-containing vacuole. The bacterium actively changes conditions within the CCV to create an acidic environment favorable for its growth. It also secretes effector proteins through a type 4 secretion system, necessary for forming and maintaining the CCV. These proteins disrupt host cell processes to increase the acidity of the vacuole and block the action of lysosomal enzymes that could destroy the bacteria. The pathogen's ability to inhibit phagolysosome fusion is a key virulence trait for immune evasion. This allows the bacteria to avoid being destroyed by the host's immune system and establish a chronic infection. Coxiella burnetii can be shed in milk, urine, and feces of infected animals, as well as through birthing fluids, facilitating its spread to new hosts. The disease manifestations can range from flu-like symptoms to severe and chronic conditions such as endocarditis. Its intracellular persistence can lead to chronic Q fever, which is difficult to treat and can cause long-term health problems. Pathogens can adhere to and invade host tissues, evade host immune responses, persist within the host, and exit to spread to new hosts. They share similarities with other pathogens, particularly in their ability to adapt and evade the host immune system. These traits are commonly seen in many successful pathogens and are necessary for their life cycle and propagation. The unique features, such as the production of mycolactone by the Mycobacterium ulcerans and the intracellular survival of the Coxiella burnetii, are what set them apart. These characteristics contribute to the pathogenesis and clinical presentations seen in the diseases that they cause. For more information, please visit any of the references listed below.